All right, let's jump right into this explainer. Today, we are decoding the absolute, incredible mechanics of tree seed propagation, taking us all the way from those high, swaying branches straight into the high-tech storage lab. Think about it for a second. Every single towering forest you've ever seen begins with this tiny, highly vulnerable package of genetic material. But actually getting that seed from a branch into the ground and making sure it survives, that is an intense battle against gravity, hungry animals, invasive fungi, and honestly, time itself. So today, we're going to explore the meticulous science and the really rugged field tactics required to collect, process, and preserve these seeds.
Here is our roadmap for the journey today. We're looking at reading the maturation signs, mastering our ground collection tactics, extracting and cleaning the seeds, waking them up from dormancy, and finally, storage and survival strategies. Let's get to it.
Now you might be thinking, why not just wait for gravity to do its thing? Why not just let the seeds fall naturally? Well, waiting usually just isn't an option. Trees have this incredibly frustrating habit of producing their absolute best seeds on the uppermost, outermost branches. If we just stand at the bottom waiting to catch them, those seeds might get blown miles away by the wind, shattered by the fall, or snapped up by hungry animals before they even have a chance to settle into the dirt. We've got to be proactive here.
Which brings us to Section 1, reading the maturation signs. Because in this field, timing is quite literally everything. It all comes down to this incredible biological mechanism called the abscission layer. You see, collecting premature seeds is kind of like trying to hatch an unfertilized egg. It just won't work. We need to know exactly when the tree is ready to let go, and the abscission layer is the signal. It's a special layer of cells that literally snaps when the seed is biologically ready to leave the mother tree. While the fruit is developing, the tree holds onto it super tightly. But once that seed is ready for the real world, this exact tissue layer dries out and weakens. That means a strong gust of wind, or, a clever seed collector can easily snap it right off.
But before we even unpack our gear, we've got to play detective in the field. We're looking for vital sensory clues so we don't accidentally harvest premature seeds that have zero chance of surviving. You want to look for obvious visual shifts, like dry pods starting to split open, or fruits turning these bright, conspicuous colors that are specifically meant to attract animals. You can give the fruit a gentle squeeze to see if it's softened up. And whatever you do, don't ignore the locals. Active foraging by native birds is a huge green light, because birds generally only pick fully mature seeds. And if you're still not sure, you can do a quick cutting test right there in the field. Slicing the seed open should reveal a firm, whitish embryo, not some gooey, milky mess.
Okay, Section 2, Mastering Ground Collection Tactics. This is where we get to step in and actively accelerate that natural fall.
Since trees are, as we established, pretty great at hiding their seeds way up in the canopy, we deploy a very specific, actionable ground collection toolkit to bring them down. Sure, you could just use leaf rakes for sweeping up whatever drops naturally, but you're going to end up with a ton of extra debris. A much better tactic? Laying out fine mesh nets and massive tarpaulins right underneath the canopy. To actively coax the seeds down onto those tarps, we use telescopic poles with hooks to snap branches under 5 meters. But for the really tall, stubborn trees, that's where we break out the heavy-duty weighted throw lines.
This is such a cool process. Using a throw line, you can systematically shake massive yields of fruit right onto your tarps. Here's how it works. You toss a thin line over an outer fruit-bearing branch, maybe 10 meters up. Then you use that thin line to haul a much thicker, sturdy rope over the branch. Once you're secure, you pull with this vigorous, rhythmic movement that we call japing. Because the abscission layer has already weakened the fruit's attachment, this shaking action effortlessly snaps the mature seeds off. It literally rains seeds down onto your carefully placed ground nets. It is incredibly efficient.
Moving to Section 3, Extracting and Cleaning Seeds. Time to remove the pulp and get these seeds ready.
So our tarps are completely full of seeds, twigs, and leaves. The outdoor battle is over, and the meticulous processing phase begins. Soft fruits are literally designed by nature to be eaten. So if we don't manually strip away that soft, fleshy pulp, the fruit is going to rot, ferment, and ultimately kill the seed inside with toxic alcohol byproducts. In the wild, an animal eats the fruit, their stomach digests the pulp, neutralizes the chemical germination inhibitors, and they safely void the clean seeds somewhere new. Well, in the lab, we basically have to play the role of the animal's stomach and artificially simulate that exact digestive process.
And depending on how delicate the seed coat is, or how sticky the pulp happens to be, we have a whole array of pretty creative solutions for this.
For small batches, you'd be surprised. Household blenders with blunt blades or even standard potato peelers work great to mechanically rub the pulp off. For commercial yields, we upscale to heavy-duty Diapig cleaners or coffee depulpers, or even blast them with high-pressure jet cleaners inside wire baskets. Got incredibly stubborn, sticky pods like tamarind? Stirring the seeds in water mixed with wood ash does the trick. And if absolutely all else fails, we can literally feed the pods to goats and let their digestion do the heavy lifting. Hey, collecting the droppings works.
All right, Section 4, Waking Seeds from Dormancy. It's time to break that stubborn slumber.
Now that the seeds are beautifully clean, we have to wake them up. This state of rest is called dormancy, and it is easily one of the most frustrating things you'll encounter. You can plant a perfectly healthy seed, wait weeks, and absolutely nothing happens. Now, imagine staring down a 5-hectare planting project. You are definitely not clipping 20,000 thick seed coats by hand. Instead, we rely on bulk chemical and thermal solutions. If a super thick seed coat is physically blocking water, we can literally shock it with boiling water or give it a highly controlled 10 to 15-minute soak in sulfuric acid. If the seed has chemical inhibitors, we leach it in continuous running water. And if it's waiting for a specific season, a physiological dormancy, we can trick it using cold stratification or soaking it in commercial plant hormones like gibberellins. But before we can actually put them in the vault, we have to understand a stark contrast in seed biology. This is a critical fork in the road. On one side, we have orthodox seeds. These guys are the cooperators. They're like hibernating animals. They let us dry them down to 4 to 7% moisture, which pauses their life functions entirely so we can store them for years. But then, on the other side, we have the desiccation-sensitive seeds.
These desiccation-sensitive or recalcitrant seeds, they are a completely different story. Long-term storage is basically impossible. We usually just have to spread them out in a cool place, spray them with water, and get them into the ground fast. Coming from moist, humid forests, they never evolved the need to dry out. They view falling from the tree and sprouting as one continuous, non-stop sprint. If you try to dry them down past 10% moisture, their metabolism shuts off and they simply die. We can slow them down a tiny bit by dropping the room temp, but make no mistake, the clock is always ticking on these ones.
Finally, Section 5, Storage and Survival Strategies. This is how we preserve the harvest for the long haul.
25%. Think about that number for a second. That is the staggering potential mortality rate for seeds in nurseries and fields. It really emphasizes the massive stakes here. Leaving orthodox seeds on a shelf just won't cut it. They are packed with nutrients, meaning everything from fungi to rodents wants a piece of them. Every single seed lost to rot or insects in storage means exponentially more effort wasted down the line.
So, to defeat the fungi, the rodents, and the microscopic beetles, we use military-grade precision to keep our vaults secure. First, we strictly control the moisture, drying orthodox seeds to that 4 to 7% sweet spot to stop them from producing toxic metabolic waste. Then, we instantly chill the room to exactly 5 degrees Celsius. We install heavy physical wire mesh so rodents can't gnaw their way in. And for the microscopic threats, like those nasty bruchid beetles that actually breed inside the seeds, we use water flotation. We float the harvest in water, and the hollow, insect-infested seeds just float right to the top where we can skim them off. Then, to guarantee no surviving insects can breathe, we seal the seeds inside carbon dioxide bags. Finally, the whole shebang goes into airtight containers so no moisture or fungal spores can ever creep back in.
Seeing all this come together really leaves us with a powerful thought. From tossing ropes high into the canopy and violently shaking down a harvest onto our ground tarps, to chemically breaking down thick seed coats and sealing everything inside high-tech, climate-controlled vaults, every single step is a total triumph of biology and human ingenuity. The towering canopies of tomorrow are sitting right there in the palms of our hands. Thanks so much for joining me on this explainer. The next time you're out walking through a forest, take a moment to look down at the ground and ask yourself, how many future giants are quietly resting right beneath your feet?